If you are taking AP Biology, you're in the right place. I'm here with Tiffany from Miss Jones from AP Bio Penguins. How are you doing today?
I'm wonderful. How are you doing today?
I'm great. I'm curious about cells. I woke up today and I said, what about cell structure? I want to know about eukaryotes and prokaryotes and Golgi apparatuses and endoplasmic reticula. I want to know about, is that what is in this unit, right? What are the big highlights for Unit 2 in AP Biology?
Of course. Oh, so you're asking me. So
Yeah.
this is just talks about the cell structure. It has to do with membrane transport, kind of cell size, and kind of that type of thing.
Well, amazing. So, Tiffany, I'm going to turn it over to you. Before I do, one thing I will say is if you don't currently follow AP BioPenguins on Instagram, on TikTok, if you're not on her webpage, I'm going to post that in the description of this video. Definitely check out all of the incredible resources. What are the big resources you have on your AP BioPenguins website?
So I have a bunch of quizzes games. I got over 60 quizzes games. I've got TikTok review videos. I've got PowerPoints. I post my daily questions on my website. I've got all the old Marco Learning sessions that I've done linked for you and everything ready to go. So anything you need is probably on my website. Oh,
Amazing.
biggest thing. I have a 316-page review guide that breaks down the entire CED, in which there are I can statement questions, there are topic questions, there's practice multiple choice and free response questions, all with answer keys. Well, the multiple choice and the free response all have answer keys for you. So all good
It's
to ready to roll and study.
so your free guide is only 316 pages. Is that all?
Only just 316. It was only 245 last year. So I think that I'm doing pretty good by only making 316.
It sounds like you are. So everyone, again, definitely subscribe to AP Bio Penguins on those channels like Instagram and TikTok. Join those quizzes games. Grab your 5 million page guide. Tiffany's going to be our guide through cell structure and function. So I'll let you share your screen and get things started.
1 second, John. I somehow lost the screen. Give me 2 seconds. Okay, got it, y'all. I'm so sorry. Okay, so share my screen. So hi, welcome. So as John mentioned, I'm Tiffany Jones from AP Bio Penguins. So with this, I always wondering, well, why are y'all penguins? Like, what is the reasoning for that? Why do I call y'all penguins? And it has to do with the fact that penguins are in their little tuxedos. They're dressed for success. So now that you're in my live, you are all an AP Bio Penguin. So first thing, what's today's session about? So first thing, I want to give you a shout out, right? So in your chat, you're going to notice there is a Miss McClinton. She is another AP Bio teacher in the country. I think she's up north somewhere. But so she is going to be my chat moderator. So if you have a question as I'm going through this, you're more than welcome to type it into the chat, and she's going to answer your questions for you as we go through it. And then we'll also have a Q&A session at the end if you want to hold any questions. So the different things we'll do, we'll talk about cellular organelles. And so we'll talk about all the different parts of the cell that are directly out of the CED. We'll talk about surface area to volume ratios and kind of the calculations you're going to need to do for that. We'll talk about membrane transport, so how materials are going to move across the membrane. I've got 2 multiple choice questions and then 1 free response question that we're going to go through just to ensure that we can kind of make a direct connection to the content. So let's get started. So cellular organelles. And I've organized these slides so that they're designed as like a 1-pager. So you can print out these slides and they're nice and organized. You could use these to study later on. And the whole PowerPoint will be available on my website, as John mentioned earlier, apbiopenguins.weebly.com. They're all there for you. So our first organelle we want to talk about is our nucleus. And so I've formatted this in a very similar way, in which I do the structure and then we'll talk about function. So the structure is a double membrane. There's a double membrane that surrounds your nucleus, and it has these little pores in it. Okay, those pores are going to allow for materials to leave that nucleus. So we know that the mRNA is, of course, synthesized inside of your nucleus. So of course, the mRNA is going to need to leave the nucleus in order to move out to the ribosomes that will be in our cytoplasm. We also see that the nucleus is where we're going to find our genetic information. Our DNA will be stored within our nucleus. And then ribosomes will be stored inside the nucleus, and they'll move out through that nuclear pore out into the cytoplasm. we have the roughy R. And so you've noticed I'm kind of circling them on the slide. So you can kind of see this little structure over here, and I keep circling them. So the roughy R is a membranous structure, and it's studded with ribosomes. Now, a common misconception is that the roughy R always has these ribosomes, right? These ribosomes will attach and detach depending on whether they are synthesizing a protein. Okay, so all of our ribosomes start out in the cytoplasm, and then they will attach to that roughy R when they have the signal. There's a little signal peptide that tells it to go to the roughy R, and that's what's going to allow it to start making that protein on that membrane. So it's a membrane studded with these different ribosomes, and the job is that it's going to make membrane-bound proteins. So proteins that are actually sitting in membrane. And this is a job for, like, if you think about glycoproteins, you think about transport proteins, you think about any proteins in your membrane. There's some that act as enzymes. There's some that help with cell-cell communication. So any membrane protein is made in that roughy R. We also see that proteins for secretion are made here. So this would be if you're secreting material. So like insulin. Insulin is going to be secreted from your cells. And so it has to be made within a roughy R. So it's surrounded by membrane. So it'll be able to go through a process called exocytosis to excrete it out of the cell. So we also see that the roughy R is in job for cell compartmentalization. I have a very hard time saying that word. That's why my eyes kind of did that. So this is just meaning that we're breaking up the cell. We're having these different locations for specific functions inside of our cells. It has a job for mechanical support. It also has a job for our intracellular transport. Thinking about that after a material leaves our roughy R, it's then going to move to the Golgi, and then that Golgi will then package and modify it for different parts of that cell. So it's part of our intracellular transport, and it's a key role in that intracellular transport. So our smoothie R, as you see up here, is also going to be attached off of that, the nucleus. It's a connective part of that membrane. And it's folded, it's tube-like, it's made up of these different structures called cisternae, okay? And it's got a couple different functions. So the big thing about it is it's for detoxification. So it'll help to break down any type of toxins within the cell. It's also going to have a job for calcium storage. So if you think about your muscle cells, your sarcoplasmic reticulum is a type of smoothie R. And so it's going to hold that calcium, and then it's going to release that calcium when it gets a signal. The calcium then acts as a secondary messenger to allow for that signal transduction within the cell. So it being a calcium storage is an essential role that the smoothie R makes, okay, or it has. We also see lipid synthesis. So it's going to make the different lipids, so like the different steroids that will be secreted out of your cell. So big important stuff that smoothie R does. We also have our Golgi complex. Now, you've seen these Golgi complex as different names. You've probably seen Golgi bodies. You've seen it as Golgi apparatus. And the CED calls it the Golgi complex. Okay. And so what's going to happen here is that it's going to just be this balancing, right? And so you can see that it's made up of these little stacks. So there's this little stacks that kind of are on top of each other. You've got your cis side that's closest to the roughy R, and you have your trans side that's on the farthest side from the roughy R. And so materials are going to come in through these little vesicles. So our roughy R made the material, these different proteins, and it's going to surround it by a membrane called a vesicle. The vesicle is then going to bind to the cis side of your Golgi. And as it moves through the Golgi is going to allow for the folding of these different proteins, and you have a chemical modification that needs to be done. So for example, insulin. Insulin has to be cleaved. It has to be cut and then rebound together in order for it to be functional. So that's an important job that's happening within our Golgi. And it's also going to package that protein. Okay, so if we're making hydrolytic enzymes, those hydrolytic enzymes are going to be surrounded by that membrane and sent out into the cytoplasm to form our future lysosomes. Or if we're making something for secretion, surrounded by a vesicle, it's then going to fuse with the plasma membrane to release it. So it's kind of, as you think back to middle school, it's our mailman or mail person in terms of our cell. It's going to package everything. It's going to distribute it to its appropriate locations and kind of send it to where it needs to be. And then we have our ribosomes, which we've mentioned them before. So ribosomes are composed of rRNA. rRNA is your ribosomal RNA. So if you remember, there's 3 different types of RNA. We have the mRNA, that's your messenger RNA that we mentioned earlier, that's going to be synthesized in your nucleus and leaving the the nucleus to go for translation. We also have our rRNA that's going to be making up our ribosomes. And then we have that tRNA that we'll talk about later when we talk about translation, which transfers the amino acids to that ribosome. So our ribosome is made up of the rRNA, plus it's made up of protein that kind of makes a large subunit and a small subunit. And we'll talk more about the functions of all those different components of the large and small when we get into translation. For now, just understand that the ribosome's job is to make proteins. That is its big job, is just protein synthesis. And there's two different types of ribosomes. We have bound ribosomes and free ribosomes. Our free ribosomes are going to be found in our cytoplasm, and they're just free floating. And there'll be a small subunit, a large subunit, and they'll attach around the mRNA. And then once they get the signal, then they can move to either the nuclear envelope or to the rough ER to continue that protein synthesis. So bound ribosomes are attached to rough ER, and then our free ribosomes are just in cytosol. And the difference of location does tell us a difference of their function of protein, right? So if it's a rough ER protein, it's a bound, if it's a rough ER ribosome or it's a bound ribosome, then that means that it's going to make a membrane protein or protein for secretion. If it's a free ribosome, it's going to be making a protein for the cytoplasm or just some type of protein that is going to be in the cytosol. So I hope that has been helpful so far. Thank you, Ms. McClendon. I see you're answering a whole bunch of questions in the chat. You're doing a great job helping me out with that. So we're not done with our cell organelles. We have way more to do. So this one's going to be formatted a little different because I wanted to show you the real big structures of these things. So this is our mitochondria. So our mitochondria are going to be, there's a couple parts to it. Okay, so you have a double membrane, right? So there's this outer membrane here that tends to be really smooth, right? And then you're going to have an inner membrane. This inner membrane called your, my gosh, my mind's blank, cristae, is going to be highly folded. Okay, so this highly folded allows for an increase to surface area. The more surface area we have, the more locations we have for the different roles and the jobs that take place within our mitochondria. And so here we see that there's, of course, ribosomes in it. You'll remember from endosymbiotic theory that the mitochondria does have its own ribosomes, which means it can synthesize its own proteins, and it has its own DNA. So it's kind of like a really cool structure that this mitochondria is. So big things, it has a double membrane, outer membrane is smooth, inner membrane is highly folded. The functions, okay, you're going to find that this is the site of oxidative phosphorylation. So you'll see that we have the electron transport chain sitting within this inner membrane. We have the electron transport chain sitting within that cristae, okay? And as those electrons move down their energy level, they're going to pump protons. So they pump it out into this inner membrane space. So we're going to see that there's this compartmentalization that takes place within our mitochondria, that it can allow for different cellular processes to take place at different parts of the mitochondria. And so we also see that we have the matrix. The matrix is this inner part, like on the inside of the cristae. And in that matrix is where we're going to see the Krebs cycle taking place. So the Krebs cycle is the second step that we'll see in cellular respiration, which will be at our next thing. But this is where we will continue the breakdown of our glucose. So we've completely broken down the glucose, and we've made all those NADHs and the FADH2s and all of the amazing stuff that's going to move into that last stage of oxidative phosphorylation. So let's get rid of the mitochondria. Let's talk about a chloroplast. Okay, so a chloroplast is also going to have a double outer membrane. So it actually has two membranes out here. There's an outer membrane and an inner membrane. And then inside of there, you're going to see these little stacks. These stacks are called grana, and then they're made up of these little sacs called thylakoids. And so these little thylakoids are stacked up together, and in the thylakoid membrane is where you're going to find all the pigments for photosynthesis. So you'll see that we have the chlorophyll. You'll see this is where our photosystems are going to be, remembering that the photosystems have the chlorophyll, the reaction center complex, and of course they have an electron transport chain for us to synthesize ATP in that chloroplast. And so we also see that it has this stroma. So there's a jelly-filled area, which is the cytosol of the chloroplast. And so we'll see the light reactions taking place within the thylakoid. And then we'll see the Calvin cycle taking place in that stroma or in that liquid area. So I think I've got everything there. So getting rid of our mitochondria and our chloroplast so that I can make the finished part of the pretty diagram. So lysosome, I think I've mentioned that before. Lysosome was synthesized as the hydrolytic enzymes were made by the rough ER that were then packaged by the Golgi. So all it is a little sac that has hydrolytic enzymes. And if you remember back from unit 1, hydrolysis is the act of breaking something down. So it's how we are breaking up our macromolecules, our polymers, into monomers, and they will break water molecule. And by breaking that water molecule, we can, of course, break that bond. And so these hydrolytic enzymes are going to be responsible for digestion. So we have intracellular digestion taking place. So this can recycle any type of organic material within the cell. So if a protein misformed, we can break it down within the lysosome to reuse those amino acids. If there's an organelle that's non-functional, it can be broken down by a process of autophagy to break down that organelle that's no longer functional. We have a programmed cell death, so apoptosis. So the cell will have an autocrine signal that will tell it to break down. We'll have nucleases that break down the nucleic acids, and then we have proteases that break down those proteins, and then the lysosome will kind of help to continue that breakdown for us. So yay, lysosome. And then we have our vacuole. Our vacuole is just a membrane-bound sac. It's really what it is. It has a lot of different functions. It's for storage. It can release macromolecules. It's for cellular waste. There's a ton of stuff that happens in your vacuole. And those vacuoles there's 3 main ones that we focus on. You have your central vacuole that we find within plants. And its big job is water retention. This is what's going to give you that turgor pressure, that the fact that there's all this pressure pushing onto the cell wall that kind of keeps that plant cell erect and kind of firm. And so it's because of the central vacuole that has that. It also has a couple different other materials that are stored within that central vacuole. But the big thing about the central vacuole is that it's for water retention. It's kind of to store all that water so that it's part of osmoregulation. We also have our contractile vacuation, which is also contractile vacuole. It's also part of osmoregulation. And so within this, we're going to see that we've got these protists, right? It's a freshwater protist. And as we know, in fresh water, water rushes into the cell or the organ or the organism. And so as that water rushes in, if too much water rushes in, it's going to burst, which we'll talk about in a little bit. That just has to do with the hypotonic environment versus the hypertonic. And so the water rushes in, which is problematic for that protist. So that's a contractile vacuole. The water goes into the contractile vacuole. Contractile vacuole then contracts and it pumps that water out. So it's a way that it can ensure that the cell doesn't burst, or this unicellular protist doesn't burst from this water rushing in. We also have a food vacuole. The food vacuole is formed by phagocytosis. Okay, so it's going to go through, and this is cellular eating. It'll engulf some type of food material, and as it engulfs that food material, it then forms a membrane around it. So that's now a food vacuole. That fuses with our lysosome to allow for digestion. And this is what you'll see happening in your macrophages or your white blood cells within your body. They will break down bacteria and other pathogens that are inside of your body that need to, protect you, right? And so it said before they fuse with the lysosome. So, that is the big organelles that are directly in the CED. I went through the CED last night and I went through and I found every single organelle that it mentioned. Those are the big ones that we have in there. If you see something else, then you'll probably be giving some type of information about it. So they may talk about peroxisomes, but again, they have to give you information about them. So the questions I'll ask you aren't going to be point blank questions about the organelles. They're going to be application questions. So they'll tell you I don't know, there's this disease called Tay-Sachs. And with Tay-Sachs, you're going to have a buildup of lipids in the brain of the organism or the person, okay? And so what organelle has a problem? Well, you would then think, okay, well, it must be the lysosome, because the lysosome is unable to break down that extra lipid material. It has an error in its hydrolytic enzyme. So you have to make applications to these, okay? They're not just going to be straight questions saying, what's the function of the lysosome? So you have to think about application to these things. So cell, right? So we have all these different types of cells, and these cells have different shapes, right? So one of the things that you're going to have to do, oh no, it's math. You have to be able to calculate the surface area. So the space on the outside of the cell versus the volume, which is the space on the inside of the cell. So there's 4 different equations you'll see on the equation sheet. So we have the surface area and volume of a sphere. Okay, so you know, of course, here's the volume, here's the surface area of it. You would have to know how to calculate the surface area and volume of a cube. So again, here's your equations. They're very simple, just sub them in. So for example, here's the side length. You'll just sub it straight in and cube that. And then you'll do 6 and then the side length squared for the cube. Here we see the rectangular solid. And that will just be a different length, width, and height. Same thing, you're just subbing them into the equation. And then here we have a cylindrical solid, or just like a cylinder. And so what they can do is they can either A, give you surface area and volumes and then ask you how they compare, or they can ask you to calculate the surface area volume ratio of 2 cells, or they can kind of vary it slightly, maybe give you cells that have already formed, and you have to then decide which one has the greatest surface area to volume ratio. But why do we care about this? What is the reason for needing to calculate this? So when we have a small cell, that small cell is going to have the highest surface area to volume ratio. Now that's important. Why? Because the membrane is where all of the nutrients are coming in. That's also where all the wastes are coming out. Versus the volume is all the space on the inside. And if it takes a long time for materials to get to the membrane, to get out of the cell or to get into the cell, that's going to be a problem. So if you think for a second about being in like this huge auditorium, right? And you're like in a big concert or something, there's all these people around, right? Versus being in a classroom. If you need to get out of that cell, you need to get out of that space, it's going to take you a very short amount of time to get out of that room, that classroom, than it would for you to get out of a big auditorium. And that has to do with that surface area volume ratio. So the most efficient, the best cells you can have are your small cells, because they have this big, large surface area to allow for more stuff to get in and out, but they don't have a lot of volume. So things can move throughout the cell very quickly and efficiently. And I hope that made sense. So if you want me to go through math problems on that, I believe that there's a marker learning session that I did last year where I went through calculations for every single equation on your formula sheet. And that is linked on my website, so you can kind of access that if you need to. So membrane transport. Wait a minute. How are we talking about membrane transport if we haven't even talked about the membrane? So let's first talk about the membrane. So here is a graphic of our plasma membrane. Plasma membrane is made up of course, phospholipids. So here you see we have our phospholipid. It's got the hydrophilic head and it's got these fatty acid tails. And the way they're set up is that you've got, of course, the head facing outward and the tail facing inward. Okay. And that's making a bilayer, where there's 2 layers of phospholipids that face one another. And so it makes this hydrophobic interior core. So this whole part right here is hydrophobic. And then on the outsides of it, so on the extracellular side and intracellular side, is going to be hydrophilic because, of course, everything is bathed in water, right? Water is everywhere. We also have these things called proteins, right? So you're going to have peripheral proteins that kind of just sit on the membrane. You also could have transmembrane proteins that kind of will go through. These can have things like channel proteins. These can give you carrier proteins. So there's all these different things that can happen with these different proteins. We also have glycolipids. Glycolipids are going to be a carbohydrate, a very short carbohydrate attached to a lipid, usually a phospholipid. So we have this kind of little carbohydrate. We also can have the same thing where there is a short carbohydrate attached to a peripheral protein. And those have the job of cell-to-cell communication. So this allows your cells to, of course, know whether they're hosts, cells, so if you've had a pathogen that comes in your body, you need to be able to recognize the glycoproteins on your own cells versus pathogen glycoproteins. This can also allow for cell-cell communication in terms of binding. So like your helper T cell is going to bind to the B cells to activate them to make the plasma cells, or bind to the cytotoxic T cells to tell them to start to break down pathogenic, like the cells have been taken over by a pathogen. So there's this communication that takes place within these glycoproteins and glycolipids. Another example would be like red blood cells, like your A blood cell and your B blood cell. They have different glycolipids. And I could be misspoken that. It could be glycoprotein. I think they're glycolipids. Anyways, so they have different glycoproteins, glycolipids on them that can tell you the difference between an A cell and a B cell. So these are really important for cell-cell communication. We also have cholesterol. So cholesterol is going to sit in your plasma membrane, and it has a job to inhibit constriction, right? So if it gets really, really cold, things start to condense and they get really close together, right? So we don't want our membrane to get solid, right? We need our membrane to stay fluid. So we have these fatty acids, we have these cholesterols in there to inhibit those from getting too close together. And the opposite is true. If it gets really, really hot, things start to space out and they move faster apart from each other. So we don't want these big gaps and these big spaces in our plasma membrane. So the cholesterol acts as a buffer. It helps to keep the membrane fluid and it helps to keep the membrane secure, okay, to ensure that we have this. So I think that we've gotten everything that's there. Let's talk about membrane transport. So we have simple diffusion. Simple diffusion is a type of passive transport. It requires no NRG. I shorthanded that. Ha ha ha. Okay, never mind. Anyway, so there's no energy involved there, right? This means that things are going to move down the concentration gradient. So thinking about the fact that if you're on top of a hill and you're going down the hill, right? You have all this potential energy that allows you to move down the hill. So you don't have to use any energy to ride a bicycle down the hill, right? The same thing applies here. If we have materials that are in a higher concentration on one side of the membrane and a low concentration on the other side of the membrane, they can naturally move across that membrane down that concentration gradient, okay? So with simple transport, this will be your small non-polar item. Why does it have to be small non-polar? Because it has to be able to fit in between these phospholipids. Why does it have to be non-polar? Because this core right here is non-polar, okay? And so we need it to dissolve. It needs to be able to fit into that area. So good examples we'll see, calcium, carbon dioxide, sorry y'all, carbon dioxide, oxygen, nitrogen, those can all easily pass because those are non-polar materials, non-polar molecules. We also see that steroids can easily pass that membrane by simple diffusion, okay? So that'd be like your testosterone or your estrogen. Those can easily pass directly through because we know that our steroids are going to be non-polar. Now, it's really interesting, and I didn't know this until I looked at the CED, but a very small amount of water can actually leak through the membrane. So you can actually see a little bit of water moving through. It's not going to be a very fast process due to the fact that we're going to see that it's kind of inhibited due to the fact that this is a non-polar interior, but it can kind of leak through a little bit. So that is simple diffusion. The other type of passive diffusion we have is called facilitated diffusion. Okay, this is also passive transport, doesn't require any energy, and it's also moving down its concentration gradient. Okay, this will be just small molecules. They're oftentimes going to be polar, they'll be charged, so they'll just move down their concentration gradient. But the thing is, because they're polar, because they're charged, they can't really get in between the phospholipids. So what we have to do is we have to use transport proteins. So there's two different types of transport proteins. You have carrier proteins and you have channel proteins. Our channel proteins are just going to be exactly like this. There's a hydrophilic channel on the inside that the material can bind to and move straight through. It'll just flow through, just kind of like a channel that you see anywhere. It just flows right through. It's like a little tunnel, if you may. Versus the carrier protein is going to bind, which causes a conformational shape change. And as it binds, giving that conformational shape change, it's then going to move it across the membrane. So if we have this material, which that's probably not a good example for me to use, but if we have this material, right, and it binds, right, anytime you bind to a protein, you have a conformational shape change. It changes shape. So as it changes shape, it moves that material across that membrane. And then so that can allow us to see that carrier is going to actually carry it across the membrane. So examples, we'll see water moving through a channel protein. You'll see sodium and potassium. They have channel proteins. They also have carrier proteins. You'll see calcium moving through. And so there's a lot of stuff that can move through this. Now, there was a free response question a couple of years ago that you need to kind of be aware of the fact that these are specific, right? So if you have a sodium channel, the only thing that can move through it is a sodium channel. If you have a potassium channel, the only thing that can pass through is a potassium molecule, right? And the reason why is because it's the right size and it has the appropriate active site to bind to these materials to allow for them to pass through this area. Whew. Now, both of those I said are both types of passive transport. So we oftentimes call both of these diffusion, but simple diffusion and facilitated is just looking at whether there's a protein or whether there's no protein. Whew. So active transport. This means that you have to apply energy. You have to put something in, right? This is going against concentration gradient. An example of a carrier protein would be a sodium potassium pump. That would be a carrier protein. So when we think about active transport, we're thinking about moving against their concentration gradient, right? So we're at the bottom of that hill. We have a low concentrate of our material, and we need to add energy to go up, to go to a higher concentration area. So we're going against our concentration gradient now. This requires a transport protein. Traditionally, we see this as a carrier protein because it's going to have to bind to pump it. So we have proton pumps, you have sodium potassium pumps. Like these are things that are going to pump things across a membrane. And when I say proton pump, I mean it's an H plus. I think you know this, but just in case. And so we see that, you know, in co-transport options where like we'll bind to a molecule with that sodium, I'm sorry, not sodium, but potassium. Protons will bind with like a glucose to pump things against their concentration gradient to bring that glucose in through a symporter. Because we have a lot of stuff that happens with that active transport. And again, we have to apply some type of energy. Now, we have another thing we have. This is bulk transport. Bulk transport is a type of active transport. Okay, this is something that's way too big to move across the membrane. It can't go through a transport protein. It's just big. Okay, and so what we're going to see here is there's two effects: endocytosis, which is going to be moving things into the cell. This is how we're going to import materials, bringing them into the cell. There's three different ones. You have phagocytosis. Phagocytosis is cellular eating. Okay, so as we said before, that macrophage is going to engulf something. That's phagocytosis, cellular eating. And so when it brings it in, it makes, of course, that food vacuole. You can see here in this picture, we've had this little pseudopoda. They extend out. They surround that microorganism, the bacteria. They surround the cell debris. They surround something that we're bringing into the cell, which then makes this little food vacuole. And we talked about before, the food vacuole that will then fuse with a lysosome to digest it and break down that material. We have penocytosis. Penocytosis has to do with cellular drinking. And so a student my very first year said, you know, penocytosis, it makes you pee, no, when you drink a lot. So penocytosis, cellular drinking, which is penocytosis. And so here what's happening is it's gulping extracellular fluid. So we have like a little invagination to bring in some of that extracellular fluid. A lot of times there are solutes that are in that fluid that are brought into the cell. But for the most part, we're literally just trying to bring in that extracellular fluid. And then the last one we have is receptor-mediated endocytosis. So this has to do with when there's going to be a ligand. The ligand is going to bind to a protein on the membrane. So that's another thing. Receptor is another example of a membrane protein, which will then cause this invagination, makes this little pitted structure. And so we can bring in specific materials from the extracellular membrane, those ligands to be brought into the cell. Now, the other type of bulk transport we have is exocytosis. This is going to be how things are going to exit the cell. So a way that we can remember that is endocytosis, EN, things enter. Exocytosis, EX, things exit. And so this is what we're going to see when we export materials. Traditionally, we'll talk about this in terms of the secretion of our proteins that we made within our rough ER. So in the rough ER, we'll synthesize those proteins. It will then go through a vesicle to our Golgi complex to package it and modify it. And then it will go through another vesicle to the plasma membrane to secrete it out. And so that would be exocytosis. And as I said before, active transport, so using those carrier proteins to pump things across the membrane uses energy, as well as this bulk transport is also going to require energy to do that, okay? So we're doing good, y'all. We're way ahead of schedule. So osmosis, let's specifically talk about water movement across the membrane. And so we have three different types of solutions. And so let me just take a moment to talk about these solutions before we talk about how water moves across that membrane. So here I have a low solute concentration area, right? So there's no solutes in this side, versus on this side, I have higher amounts of solutes. So you notice that we have these little water molecules. the water molecules are going to surround the solvent. It's called solventation. And so that's how things dissolve, is that the water surrounds these different materials, and that's how they dissolve. That's why like you can get like a super concentrated solution, is because of the fact that you've gone past the point where there's enough water molecules to surround that sucrose. And so what we see here is that of course, the oxygen, which is partially negative, is going to be attracted to the partially positive part of this solute, okay? And so we have only 4 free water molecules here, versus on this side, I have 24 free water molecules. And we looked at before that things always move passively from high concentration to low concentration. So what we see is that water is going to move from where there's a high concentration of free water to where there's a low concentration of free water. So here you can kind of see that water molecule is going to move across the membrane down its concentration gradient, okay? And so this is the direction it's going to move. The problem that students have is when they think about these different solutions, they see numbers, right? You see that this side has a 0 molar, and you see that this side is, I don't know, let's call it 4 molar, okay? And you see the bigger number, and your brain immediately goes, oh, it moves from 4 to 0. But what you're looking at is the solute concentration. So you have to think about that if there's a high amount of solutes, that means there's a low amount of solvent. There's less free water that you have available to move. And if you have a low number, that means I have a very low solute and a high amount of free water that can move. So you're going to go against your better judgment, and the numbers are going to go from a low number to a high number due to the fact that we're showing a solute concentration. So here you can kind of see that, again, looking at water potential. When you calculate water potential, we're looking at that water potential has to do with that there's a high amount of the potential for water to move. So this solution right here has a low solute concentration, but a high water potential. And this side has a high solute concentration and a low water potential, because water has a natural tendency to move from high water potential to low water potential. And so as you see here, that water is moved, and the job is to try to make the concentrations of those solutions equal. And so if you have a higher amount of solvent, then you can, of course, allow that process. So we have different solutions. Your hypertonic solution means that I have a high amount of solute and a low amount of free water. This means it's going to gain water from a hypotonic solution. So when you think about these solutions, they're in terms of each other. So if you have a hypertonic solution inside of your cell, outside of your cell is hypotonic, okay? Just like if there was someone that you're describing, you're saying, well, that person is taller than this person. You are describing them in terms of each other. So when you're writing a free response question, you always have to say the solution is hypertonic to the cell. The cell is hypertonic to the solution. You have to use those terms when you're describing them, okay? So as we said before, hypertonic had a high solute and a low amount of free water, and it's going to gain water from a hypotonic environment. Isotonic means it's equal, right? So outside of the cell and inside the cell is going to have the same solute concentration and the same free water, which means that water is going to move in a dynamic equilibrium. It moves into the cell and out of the cell at the exact same rate, okay? So there is water movement, but there's no net water movement. So it goes in the cell and out of the cell at the exact same rate. They're equal concentrations, equal solutes, okay? Hypotonic solution has to do with that I have a very low solute concentration. So that means there's a high amount of free water, right? So as I said before, things are going to move from where there's a high free water to low free water. So since the hypotonic solution has a high amount of free water, it's going to lose water to that hypertonic solution. And so we always see that the water is going to move from a hypotonic to a hypertonic solution. So it always goes from high water to low water. So as I said before, hypertonic solution is going to mean it is a very low water potential, and a hypotonic solution means it's going to be very high water potential. Water is going to move from the hypo to the hyper. Wait a minute, but now I have to connect this to an actual cell? Whew, yes, we do. Sorry. So here I have a plant cell, okay? Knowing this plant cell has this cell wall, right? And so this solution you can see has a very high amount of solute on the outside of it, okay? So because of this high solute concentration outside, this is a plant cell, and the plant cell is in a hypertonic hypotonic environment. So since the cell is in a hypertonic environment, the cell loses water. So if you notice, here's this little central vacuole, right? The central vacuole is kind of small, right? Because it's losing its water, right? So here's our cell membrane. Since it loses water, it's going to become plasmolyzed, right? The cell wall, the cell membrane is going to pull away from the cell wall because it's losing that water. The cell wall can stay exactly where it is because it's rigid, but you're actually going to see the cell membrane going to pull away. This is called plasmolysis. If we look at an isotonic environment, we said equal amounts of water going in and out. So this is going to cause your plant cell to be flaccid. It'll be limp. Okay. So it does have water, but there's not a lot of pressure. And that pressure is what gives the rigidity to the plant cell. So in this hypotonic environment, there's a very low amount of solute, right? So because the cell is in a hypotonic environment, water rushes into the cell. And as that water rushes in, we see that it kind of fills up this vacuole, right? And so it gives that turgor pressure that we need for that cell wall. So that's our plant cell. If we look at an animal cell, very similar thing, right? If an animal cell is in a hypertonic environment, it loses water, which causes it to shrivel, okay, which is very bad. Another word is crenate for that. If it's an isotonic environment, this is what's normal for a animal cell, and so this is good. It should be an isotonic environment. And then if it's in a hypotonic environment, so much water rushes in that it just bursts. Okay. And so in terms of a plant cell, yes, it's ideal for them to be in a hypotonic environment. They do want to be in the hypotonic environment versus an animal cell wants to be in an isotonic environment. So there's that. Okay. So I've got some questions for us just to ensure that we were kind of getting that quick little moment. Now I'm going to tell you that there is a 30-second delay between when I say something and when you hear me. So I'm going to give a pause just so that you can kind of think through your answers. But I'm probably going to end up talking before you actually have a chance to type in your answer. So here we see that we have a simple cuboidal epithelial cell that line the ducts of a certain human exocrine gland. Various materials are transported into and out of the cell by diffusion. They give you the formula, which apparently in my copy and paste, I didn't correct this. This should be S squared and this should be S cubed. Sorry, y'all. And so they're giving you the formulas for you to be able to calculate this. Which of the following cube-shaped cells is the most efficient in removing waste by diffusion? So number one, this is a multi-choice question, which means you have approximately a minute and a half to answer this question. I'm going to be completely frank with you. You're not going to do this calculation in a minute and a half, and you're going to waste your time if you try to do that. So you have to think about what you know, right? You know which type cell would be most efficient, right? What kind of cell would be the best? Do you want to have a large cell or do you want to have a small cell? So I'm going to pause about 10 seconds for you to try to think of what's your answer. Cool. I hope you came up with your answer. Anyway, so we would expect that 10 micrometer cell to be your best cell because it's going to have the highest surface area volume. You're more than welcome to check the math. You're more than welcome to do this calculation. But in terms of your exam, you need to find things that can allow you to speed up, right? You don't want to waste time doing a calculation that you know the content. You know that the smallest cell will be your most efficient. These are all the exact shape, same shape. So you know the smallest cell would be the best one. Oh, I didn't know that. Ms. McClendon just mentioned that plant cells will burst if their cell wall is damaged. That makes me sad. Anyways, so our second question that we have, which I apparently didn't do my editing I needed to do to not cover up that multiple choice. Sorry, y'all. Anyway, so here I have these two different solutions, and you might have done this lab with your class, right? In which I've got starch and I've got glucose inside of my dialysis tubing. I've got water on the outside. And then after we allow it to diffuse, we see that there's water and glucose on the outside, and then I see that I have starch and glucose on the inside. So they're asking you to which of the following best represents the amount of starch, water, and glucose in the dialysis bag over the course of the investigation. So we're seeing how much is in the dialysis bag over time. So I'm going to give you a moment to kind of look through that and see if you can kind of figure out which one you think it is based on what you've been given. Now, I also like to try to show y'all that you can get answers from the prompts, and that's what I'm going to show you when I talk in just 10 seconds. Cool. So if we look at this, we see that there's starch inside of the bag. And after the experiment, we see that there's starch on the outside of the bag. The starch has not moved. The starch is in the exact same concentration. It is not in the solution. The solution does not have any starch. It must have been too big and it couldn't pass through the pores in the dialysis tube. And so if we look at our key, it tells us that starch should be this little dotted line. So I can see that this first option, starch stays the same. I see the second option, starch stays the same. But here, I see that the starch is increasing inside of my bag. Well, that's not true. So we can eliminate that answer. We also see the same thing here, where the starch is supposedly increasing, which also we know is not happening because of the fact that, based on the picture, right? Use your diagrams. So now let's look at the glucose. So the glucose starts off in the bag. And after the experiment, it's on the inside of the bag and it's on the outside of the bag, which means that it must have left the bag. So if we look again at our key, the glucose is going to be the solid line. So I expect the answer to be the one that has my glucose decreasing from my bag because of the fact that it's now moving out of the bag into the solution. So, I went too fast. So this one, we see that decrease. So this I think is my answer, but let's just double check the other one. But this one shows that it's increasing the bag, which we know is not true. So we can eliminate that answer. And then if I just check the water is we see that this one is kind of, little. And then now we see that it's kind of bigger. So we know that water has moved into into the little bag. So we know that water is now increased. So my answer is this first one right here. So I hope that that helps you out. And then we have a free response question. And I see them kind of longer than I want it to be. So I'm going to rush through this a little bit. So it tells us fibrosis is a genetic condition that is associated with the defects in the CFTR protein. You don't need to know what this protein is. It's fine. The protein is a gated ion channel that requires ATP binding in order to allow chloride ions to diffuse across the membrane. Cool, they told me. In the provided model of the cell, draw arrows. So this is similar to what you'll see for your 5th question on your AP exam. You're going to have to draw something. So draw arrows to describe the pathway for production of a normal CFTR protein from gene expression to final cellular location. Now, so it's gene expression, right? So where are we making the mRNA? Well, making the mRNA inside the nucleus. So it's going to go from the nucleus to my rough ER. My rough ER has those ribosomes, we said. My ribosomes are going to allow for my proteins to be synthesized, right? Because we know that this is a protein. So then after we synthesize it at the rough ER, it's then going to move to the Golgi, right? The Golgi's job, we said, was to package and modify. And so it goes to Golgi for that packaging. After the Golgi, after the package, after the modified, it's going to, what did I do? It's going to move to the plasma membrane, okay? Now, I graded this question on the AP exam. You had to end your arrow at the plasma membrane. Why? Because it tells us that this is an ion-gated channel, right? It's going to have to be sitting in a membrane in order to do that. So if you extended your arrow past that outer part, you show that it was secreted out instead of sitting in a membrane. So do be very careful when you're drawing your diagram, okay? So the answer here should have been nucleus to the rough ER, to the Golgi, to the plasma membrane to show that pathway that we were taking. So then it says, to identify the most likely cellular location of the ribosomes that synthesize the CFTR, okay? So remember, there's 2 different types of ribosomes. We had the free ribosomes that just were in the cytosol, and we had the bound ribosomes that were on the rough ER that will allow you to make membrane proteins. So since I'm looking for a membrane protein, it has to be the rough ER. Now notice that this rough is in parentheses. That means that that is an optional answer. Like you don't have to say rough, but you had to at least say endoplasmic reticulum. And then it says identify the most likely cellular location of a mutant CFTR protein that has an amino acid substitution in the ATP binding site, okay? So I need to figure out, where does my protein end up? Well, it's mutated, right? So does that change its location? Nope, it's still going to be in the same spot. It just has a different function now. So it's still going to be sitting in the membrane. You could have said cell membrane, you could have said plasma membrane, you could have just said membrane. So with 450, that is the end. So I have this slide up here just to give me a pause moment. So now is where I was going to give you Q&A time. Now, there's about a 30-second delay, as I said before. So I'm going to show you what I have on apbiopenguins.weebly.com. And in that time that I'm showing you this, you can type your question into the chat. So I'll be able to have those questions there. So I'll be able to answer them for you if there's any questions that Miss McClinton didn't answer for you. So let me pull my penguins over here. So here I have my website. Okay, so if you go to the website, the big thing you're going to notice is that if you go right here to 2021-2022 exam review, you're going to see this picture right here. This picture will open up and allow you to get to a document. It's a Google Doc. It's 316 pages, and it has my whole review guide. Okay. You also see there's some practice exam questions there for you. And then there's this calendar. This calendar shows you what I'm doing on Instagram every day. So you know exactly what topic I'm doing every day. Also in the same spot, you see there's a TikTok review. All of my content TikToks have been exported and posted here by using YouTube. I have quizzes games. There's over 60 quizzes games that you can use. And these are actually opened up already for you, okay? There are codes that you can use, type them straight into quizzes to be able to play those games. And I've got them for each unit. I've also got, big CED ones. There's a ton of games on this website for you. So that it can kind of help you out there. Also, there are FRQ Fridays. So last year I did a whole bunch of FRQs. You can watch those videos. Here's how you would find all my Marco learning sessions. If you go here, you can access all of the Instagram reviews. They're exported as pictures. And so they're in a whole bunch of folders to allow you to be able to see all the questions that I've asked in case you don't have Instagram or in case you don't want to, see the stories each day. And there's a bunch of resources there. If you go to 2021, there's last year's daily review. So you can actually see PowerPoints of every single question that I asked on my daily review last year. So you can already have them all in a nice, pretty PowerPoint to review with. I've also got all of my Instagram live sessions that I did last year. If you need those for another session, there's question and answer PowerPoints. There's numeric responses. If you're struggling with numeric response, knowing that there's not a numeric response on the exam, but the math is still going to be in your free response questions. It'll still be in the multiple choice questions. There is a ton of numeric response practice under that 2019-2020 review. So with that, let's pop back over to YouTube so that I can see any type of questions that you have. And I don't see any questions. It looks like Miss McClinton did a great job, and you all have your questions answered. So with that, John, I think we are done.
Great. We'll wrap up here then. Thank you for coming.